Hello everyone and welcome to Chapter 22, the respiratory system. So in this chapter, we're going to be looking at the respiratory system as a whole. We're also going to look at the events that cause inhalation, exhalation, and gas exchange, as well as how oxygen and carbon dioxide are transported in the blood. So lots to cover in this chapter. So the respiratory system consists of the nose, the pharynx, which is the throat, the larynx, which is the voice box, the trachea, which is the windpipe, bronchi, and the lungs. Now, respiration is the exchange of gases between the atmosphere and then blood and cells. So there's kind of going to be two places where we see exchange of gases. So exchange between the atmosphere and blood, and exchange between blood and cells. And then we also see the opposite effect too. So between cells and then blood back to the atmosphere. Now, in order for respiration to occur, there are three processes that need to occur. First one is ventilation. So this brings air into and out of the lungs. Essentially, we know it as breathing. Then there's external or pulmonary respiration. Now, this is the exchange of gas between the alveoli of the lungs and the blood in the pulmonary capillaries. So we're kind of looking at, if I kind of number this one, we're kind of looking at this exchange right here between the atmospheric air and blood. So that's between the gas in the alveoli of the lungs and exchanging that with the blood in the pulmonary capillaries. And then our third process is known as internal respiration, also known as tissue respiration. And this is the exchange of gases between blood in systemic capillaries and tissue cells. So we can kind of label this number 2 here, and this is going to be this exchange right here. So between blood and the tissue cells. Now, as it would make sense, the cardiovascular and the respiratory system really work very closely together to bring oxygen to tissues and carbon dioxide away from the tissues. The different parts of the respiratory system can be classified according to either structure or function. So structurally, the respiratory system consists of two parts. These are the upper respiratory system, which includes the nose, nasal cavity, pharynx, and associated structures. And then there's the lower respiratory system, which will include the larynx, the trachea, bronchi, and the lungs. Now, functionally, the respiratory system also consists of two parts. These are the conducting zone and the respiratory zone. So first, the conducting zone. This consists of a series of these interconnecting cavities and tubes, both inside and within the lungs. So these are going to include the nose, nasal cavity, pharynx, larynx, trachea, bronchi, bronchioles, and terminal bronchioles. Now, really, the function of the conducting zone is to filter, warm, and moisten air and conduct it towards the lungs. Then the respiratory zone. This consists of tubes and tissues within the lungs where gas exchange occurs. So these include the respiratory bronchioles, the alveolar ducts, alveolar sacs, and alveoli are going to be the main sites of gas exchange between the air and the blood. The respiratory system has three main functions. So it provides for gas exchange. So this is going to be the intake of oxygen for delivery to body cells, and also that removal of carbon dioxide produced by body cells. It helps to regulate blood pH, and it also contains receptors for the sense of smell. It's going to filter that inspired air, produces vocal sounds, and excretes small amounts of water and heat. So like I said earlier, the structural classification of the respiratory system is the upper and lower respiratory system. So the upper respiratory system consists of the nose, nasal cavity, pharynx, and associated structures. And then that lower respiratory system consists of the larynx, trachea, bronchi, and the lungs. So first looking at the nose. So the nose is a specialized organ at the entrance of the respiratory system that consists of this visible external portion and then also an internal portion inside the skull, which is called the nasal cavity. Now the internal structures of the nose have three functions. First one is that warming and moistening, filtering the incoming air. Second one is detecting olfactory stimuli, that sense of smell. Remember, that is completed by the olfactory epithelium here. And then our third function of the internal structures of the nose is to modify speech vibrations as they pass through the large hollow resonating structures. Now, the frontal bone, the nasal bones, and the maxillae, these are going to form that bony framework of the external nose. Now, the nasal cavity is this large space in the anterior aspect of the skull that lies inferior to the nasal bone and superior to the oral cavity. The nasal cavity is going to be this big cavity in here that whole cavity there. This is going to be lined with muscle and mucous membrane. There's a vertical portion called the nasal septum. The nasal septum divides the nasal cavity into right and left sides. Anteriorly, the nasal cavity merges with the external nose, and posteriorly it communicates with the pharynx through two openings, which are called the internal nares. So this is kind of the internal nares here, which is right here. This is going to be the opening where the nasal cavity kind of turns into the pharynx, right there. The pharynx, or throat, is a funnel-shaped tube about 5 inches long, and it starts at the internal nares right here, and it extends down here to the level of the cricoid cartilage. Now, the cricoid cartilage is the most inferior cartilage of the larynx or the voice box. And we'll look at the larynx later as well. Now, the pharynx can be divided into 3 anatomical regions. These are the nasopharynx, the oropharynx, and the laryngopharynx. The superior portion of the pharynx, which is called the nasopharynx, here it's in yellow. It lies just posterior to the nasal cavity. So this is the nasal cavity here, so it lies just posterior there. And it extends to the soft palate. So the soft palate is here. This is going to be the, forms the posterior portion of the roof of the mouth. It's going to be this arch-shaped muscular partition, and it also is going to be this partition between the nasopharynx and the oropharynx. So this line right here, where the soft palate ends, is where the oropharynx begins. Now there are 5 openings in the wall of the nasopharynx. There's going to be 2 internal nares, one for the right nasal cavity, one for the left nasal cavity. 2 openings that lead to the auditory tubes or the eustachian tubes, which are right here. And then also the opening into the oropharynx. The intermediate portion of the pharynx, which is the oropharynx in blue here. Now this lies posterior to the oral cavity. So the oral cavity is going to be here. So this is actually the tongue, but the oral cavity is here. So it just lies posterior to that. And it extends from the soft palate to the level of the hyoid bone. So the hyoid bone is right there. So those are going to be kind of the areas that surround the oropharynx. Hyoid bone up to the nasopharynx, and then opening to the oral cavity. So it has kind of 3 openings, I guess. The opening to the mouth. So the opening to the oral cavity right here. It's going to be 1 opening here. And then just the opening between the nasopharynx and the laryngopharynx. Then the most inferior portion of the pharynx is the laryngopharynx. This begins at the level of the hyoid bone, right here, and ends at the cricoid cartilage. At its inferior end, it opens to the esophagus posteriorly. So the esophagus can be here, and the larynx is going to open to it anteriorly. So here we have the larynx, and here's the esophagus. Now the oropharynx and the laryngopharynx both have respiratory and digestive pathways. So respiratory and digestive. Whereas the nasopharynx only has a respiratory pathway. The larynx, or the voice box, is a short passageway that connects the laryngopharynx with the trachea. Now it lies in the midline of the neck, anterior to the esophagus, as well as it lies anterior to the 4th through 6th cervical vertebrae. So these are going to lie anterior to C4 to C6. So these are the cervical vertebrae, not to be confused with the complement proteins. Now the wall of the larynx is composed of 9 pieces of cartilage. Now 3 of these pieces occur singly, so just a single piece of cartilage. These are the thyroid cartilage, which is also known as your Adam's apple, the epiglottis, and then also the cricoid cartilage. These are the single occurring pieces of cartilage of the larynx. And then there are 3 pairs, or 3 that occur in pairs. So these are the arytenoid cartilages. So these are going to be right here. This one and this one here. I'll do a better box for that one. It's kind of right here. And then the corniculate cartilages are also appearing in a pair just on top of the arytenoid cartilage, right here and here. And then the third pair of cartilages doesn't actually show on this picture, but we'll actually see it on the next slide. And these are the cuneiform cartilages. Now, of the paired cartilages, the arytenoid cartilages are really the most important because these are going to influence changes in position and tension of the vocal folds. And the vocal folds are the true vocal cords for speech. So let's put this over here, the arytenoid cartilages. So these are the most important paired cartilages of the larynx. And this is because they influence the changes in position and tension of the vocal folds. So those are the true vocal cords for speech. Now the extrinsic muscles of the larynx connect to cartilages, or connect the cartilages of the larynx to other structures in the throat. And then the intrinsic muscles of the larynx will connect the cartilages to one another. So extrinsic muscles connect cartilages to other structures in the throat. and the intrinsic muscles connect the cartilages within the larynx to each other. So here on this sagittal section, we can actually see those cuneiform cartilages. Those are going to be those paired cartilages that we didn't see on the last picture. Okay, the cavity of the larynx. This is the space that extends from the entrance of the larynx down to the inferior border of the cricoid cartilage. Now, the portion of the cavity of the larynx above the vestibular folds, so these are labeled right here, the vestibular folds are going to be our false vocal cords. So the portion of the cavity that lies above these vestibular folds is called the laryngeal vestibule, which is labeled inside here. The portion of the cavity of the larynx below the vocal folds, this is the infraglottic cavity. The thyroid cartilage, which is here and also here, this is also known as our Adam's apple. This consists of two fused plates of hyaline cartilage that form the anterior wall of the larynx, and they kind of give it this triangular shape. It is present in both males and females, but usually it's larger in males, and this is due to the influence of male sex hormones on its growth during puberty. The ligament that connects the thyroid cartilage to the hyoid bone is called the thyrohyoid membrane. So this is a ligament. And it's going to connect the thyroid cartilage down here to the hyoid bone. That's by that ligament there. Now, the mucous membrane of the larynx forms two pairs of folds. There's a superior fold that are called the vestibular folds. These are also known as the false vocal cords. And then the inferior pair of folds is made from the mucous membrane of the larynx are called the vocal folds. And these vocal folds are going to be the true vocal cords. Now, the space between the vestibular folds is known as the rima vestibuli. Also sometimes known as the rima glottis. Now, while the vestibular folds don't function in voice production, they do have other important functional roles. Now, when the vestibular folds are brought together, they function in holding breath against pressure in the thoracic cavity, like what happens when you strain to lift a heavy object. Those vestibular folds will help to hold the breath against pressure in the thoracic cavity, kind of help you to lift those heavy objects. Sounds originate from the vibrations of the vocal folds, but other structures are really necessary for converting the sounds into recognizable speech. So the pharynx, the mouth, the nasal cavity, and even the paranasal sinuses will all act as resonating chambers that give the voice its human and even individual quality. Moving on to the trachea. So the trachea is also known as the windpipe. This is a tubular passageway for air. It's about 5 inches long and only about an inch in diameter. Now it's located anterior to the esophagus, and it extends from the larynx to the superior border of the 5th thoracic vertebrae, and this is where it divides into the right and left pulmonary bronchi. So the primary bronchi here are going to be at about T5, the 5th thoracic vertebrae. Now there are about 16 to 20 of these incomplete horizontal rings of hyaline cartilage that resemble the letter C. These are going to be the tracheal cartilage. So about 16 to 20 of these tracheal cartilage. So you can kind of see they just are kind of a C-shaped here. They're an incomplete ring. And they're going to be stacked on top of one another, but they're connected to each other by dense connective tissue. They're not fully completed or not fully a full circle of hyaline cartilage, and this is in part due to the esophagus being right here. Now, if you do eat a large meal or something large in diameter goes down your esophagus, the tracheal cartilage not being a complete ring allows the esophagus to kind of get quite large here. Allows the esophagus some breathing room in case you do ingest something that's larger. Now, at the superior border of the 5th thoracic vertebrae, T5, the trachea is going to divide into a right main bronchus and a left main bronchus. These are also called the right primary bronchus and left primary bronchus. So the right main bronchus will go into the right lung, and the left main bronchus will go into the left lung. Now, at the point where the trachea divides into the left and right main bronchi, there's an internal ridge called the carnea that's formed by a posterior and somewhat inferior projection of the last tracheal cartilage. So you can see that carnea kind of briefly right here. It's just kind of a strange shaped tracheal cartilage right where we get that border. So that carnea is kind of shaped like this, I guess, with the pipe going up there and then the left and right main bronchi here. But the carnea is going to be this kind of a strange shaped tracheal cartilage. On entering the lungs, the main bronchi will divide to form smaller bronchi. So here we can see this branching of the bronchial tree. It's going to go from the trachea, divide into main bronchi. And then those main bronchi will divide into smaller bronchi, which are called lobar bronchi. So these are also known as primary bronchi, and then the lobar bronchi are also known as secondary bronchi. Now they will have one lobar bronchi for each lobe of the lung. So the right lung has 3 lobes, and thus the right main bronchi will divide into 3 lobar bronchi. And then the left lung has 2 lobes. So the left main bronchi will divide into 2 lobar bronchi. Now the lobar bronchi continue to branch, forming into smaller bronchi called segmental bronchi. These are also known as tertiary bronchi. And these segmental bronchi will supply the specific bronchopulmonary segments within the lobes. The segmental bronchi will then divide further into smaller bronchioles. And the bronchioles in turn will branch repeatedly, and the smallest ones will branch to even smaller tubes, which are called terminal bronchioles. Now these terminal bronchioles, these contain club cells. Now club cells are these columnar non-ciliated cells that protect against harmful effects of inhaled toxins and carcinogens. And they also will function as stem cells and produce surfactant. So club cells are quite important in these terminal bronchioles. So stem cells to divide into more lung cells. And they also produce surfactant, and we'll talk about what surfactant is a little bit later. The lungs, as you can see here, are paired organs which are separated from each other by the heart, which is right here, fits into this cardiac notch of the left lung. And the lungs are also separated from other structures of the mediastinum. Each lung is enclosed and protected by a double-layered serous membrane called the pleural membrane, just labeled up here. Now, just like the pericardial membrane surrounding the heart, the pleural membrane is also a serous membrane, so there's going to be 2 layers. There's a more superficial layer called the parietal pleura. This lines the wall of the thoracic cavity. And then there's a deeper layer called the visceral pleura, and this covers the lungs themselves. Now, 1 or 2 fissures divides each lung into lobes. Both lungs have an oblique fissure. Here's one for the right lung And for the left lung here, we have the labeling at the bottom as well for right lung and left lung. So each of them has an oblique fissure. And then the right lung also has a horizontal fissure. So this divides the right lung into 3 lobes and the left lung into 2 lobes. So the oblique fissure in the left lung is going to separate the superior lobe from the inferior lobe. In the right lung, the superior part of the oblique fissure separates the superior lobe from the inferior lobe. And then the inferior part of the oblique fissure separates the inferior lobe from the middle lobe. Now on the medial view of the lungs, we can see the hilum, where the main bronchi enter, as well as blood vessels and lymphatic vessels. So at the end of our last pathway, we ended with terminal bronchioles. Now terminal bronchioles, these will divide even further into much smaller respiratory bronchioles. So we kind of go from those terminal bronchioles into now the microscopic airways, the first of which is the respiratory bronchioles. Now the respiratory bronchioles will divide into alveolar ducts. So usually each respiratory bronchiole will divide into several, maybe 2 to 11 of these alveolar ducts. And then those alveolar ducts will branch into alveolar sacs, and finally the smallest passageway in the lungs are going to be the alveoli. And the alveoli is where the actual movement of oxygen and carbon dioxide is going to occur, that transferring of molecules. So here we can see those microscopic airways. We can see the terminal bronchiole here, which will divide into the respiratory bronchioles, into the alveolar ducts, and then into the alveolar sacs and individual alveoli. So we can kind of see that we have these alveolar ducts, and around the circumference of the alveolar ducts are numerous alveolar sacs. So this entire thing is going to be the alveolar sac here. Now in alveolus, which is singular and plural is alveoli, in alveolus is a cup-shaped outpouching that's lined by simple squamous epithelium. Now an alveolar sac consists of 2 or more alveoli that share a common opening. Now the exchange of oxygen and carbon dioxide between the air spaces in the lungs and the blood takes place by diffusion across the alveolar and capillary walls, which together form the respiratory membrane. So here we can see that respiratory membrane. Again, that respiratory membrane is made by the alveolar and capillary walls. So here we can see the wall of the capillary on this side and the wall of the alveolus on this side. So Together they make up that respiratory membrane. And this is where, specifically, where we have the exchange of oxygen and carbon dioxide occur. Now, the walls of the alveoli consist of two types of alveolar epithelial cells. We have these type 1 alveolar cells. These are these long purple ones. These are way more numerous than the other type of alveolar cell. But these are simple squamous epithelial cells, and they form nearly a continuous lining of the alveolar wall. You can kind of see here just this one cell itself has extensions all the way out here, and this cell goes all the way over here and here. So it's really one very large simple squamous epithelial cell. Makes this lining of the alveolar wall. Our other type of alveolar epithelial cell is our type 2 alveolar cell. It's kind of blue fuzzy ones in this picture. These are going to be a lot fewer in number than the type 1 alveolar cell, and they're kind of found in between the type 1 alveolar cells. You can see this type 1 cell comes to here, and then this type 1 cell comes to here. So they're found in between those cells. Now these type 2 alveolar cells will also produce surfactant. Now let's briefly discuss what surfactant is. So we all remember that in the terminal bronchioles, we had those club cells that were those non-ciliated cells that produce surfactant in the terminal bronchioles. And then here in the alveoli, we have type 2 alveolar cells that produce surfactant. So pulmonary surfactant, this is it's an active complex of phospholipids and proteins. It's kind of a liquid that has both hydrophilic and hydrophobic regions. So it's a liquid that coats the alveoli as well as the terminal bronchioles. And it has several functions. So our first function of surfactant, first one is to lower the surface tension at the air-liquid interface to prevent alveolar collapse at the end of expansion. So we're going to say it lowers surface tension. To prevent alveolar collapse. So what this means is it essentially allows your lungs to expand again. It keeps your lungs, or the interior of your lungs, from sticking to each other and staying collapsed when you let all the air out of your lungs. So it prevents your lungs from collapsing, it prevents your lungs from sticking to each other on the inside, and it allows them to expand again when you take another breath. So that's going to be kind of the main big role that surfactant has. It keeps your alveoli from kind of sticking closed. It also interacts with the subsequent killing of pathogens, so helps to kind of trap and kill pathogens. Or kind of helps in that process. And also helps to regulate the immune system. But really allowing your lungs to inflate again is going to be that biggest function of surfactant. Now, associated with the alveolar wall, we also have alveolar macrophages, these little green puffballs in this picture. These are also called dust cells. And these are going to be phagocytes that remove fine dust particles and other debris from the alveolar space. Here we can see a close-up of that respiratory membrane. We can see the wall of the capillary as well as the wall of the alveoli. We have that epithelial basement membrane and then the type 1 alveolar cell. And we can see that diffusion of oxygen and carbon dioxide across that respiratory membrane. You can also very closely see that alveolar fluid with the surfactant on top of that type 1 alveolar cell too, which is very cool. Extending from the alveolar airspace to the blood plasma, the respiratory membrane technically consists of 4 layers. So our first layer within the alveolar space is going to be a layer of type 1 and type 2 alveolar cells, and also the associated alveolar macrophages, and these are going to make up the alveolar wall. Then an epithelial basement membrane underlies that alveolar wall. And then just adjacent to the epithelial basement membrane is a capillary basement membrane. And this is often fused actually to the epithelial basement membrane. And then next to that is the capillary endothelium, that internal wall of the capillary. Now the lungs receive blood by 2 sets of arteries, the pulmonary arteries and the bronchial arteries. So deoxygenated blood passes through the pulmonary trunk, which branches into the lungs. Now this is by the pulmonary arteries making up the pulmonary circulation. Now the lungs themselves also need their own blood supply, different from the pulmonary circulation. So they have bronchial arteries that supply blood to the lungs to ensure that the cells of the lungs are still able to use their own metabolism. And so they get bronchial arteries through the systemic circulation. Now, a really unique feature of the pulmonary blood vessels is that they can constrict in response to localized hypoxia. In all other body tissues, hypoxia, also known as just low blood oxygen level, causes dilation of blood vessels to increase blood flow. But in the lungs, vasoconstriction in response to hypoxia will divert pulmonary blood from poorly ventilated areas of the lungs, and it will divert that blood to well-ventilated regions of the lungs for more efficient gas exchange. So this phenomena is known as ventilation-perfusion coupling. And this is because perfusion, which is another fancy word for blood flow, the perfusion to each of the lungs matches the extent of ventilation, which is airflow. So we're going to match the perfusion to each area of the lung to the extent of ventilation to alveoli in that area. So that if there's not much oxygen coming to one area of the lungs with the air, well then we're going to decrease the amount of blood flowing to that area because that blood is not going to get much oxygen anyways. So we're going to divert that blood to areas where more oxygen is coming in through the air. So we get that ventilation, perfusion coupling. So here we have a really good table that summarizes the structures of the respiratory system. So this table is going to include just conducting structures. So it's going to include the upper respiratory system as well as the larynx and the trachea. The larynx and the trachea, remember, are part of the lower respiratory system. And then here we have the rest of the lower respiratory system, where we have both conducting structures and respiratory structures. The process of gas exchange in the body is called respiration. Now, respiration has three basic steps. So gas exchange as a whole is respiration. And there are three steps to respiration. So the first one is pulmonary ventilation. We also just call this breathing. So this is going to be the inhalation as well as exhalation of air, and it involves the exchange of air between the atmosphere and the alveoli of the lungs. So this is between atmospheric air and moving back and forth between the alveoli. Our next step in respiration is external respiration. This is also called pulmonary respiration. So this is the exchange of gases between the alveoli of the lungs and the blood in the pulmonary capillaries across that respiratory membrane. So this is exchange of gases between the alveoli and the blood in the pulmonary capillaries. So here we're going from air to air, and here we're going from air to blood. And then our last step in respiration is called internal respiration. This is also known as tissue respiration. So this is going to be the exchange of gases between the blood in systemic capillaries and tissue cells. So here from the pulmonary capillaries, it's going to travel through the whole systemic system, first through the heart and then through the whole systemic circulatory system. And so then we get the gases that's in the blood get to the organs where it's needed. So this is going to be exchange from the systemic capillaries to the tissue cells. So these weird little arrows, I'm not sure if you've seen these before, but these mean like arrows back and forth. So essentially we're always going to go have like oxygen moving this way, O2, and then we'll have like CO2 moving this way. So these back and forth arrows always just mean that oxygen is going in one direction and CO2 is moving in the other direction. So with the systemic capillaries to the tissue cells, here we're going from carbon dioxide and oxygen in the blood to interstitial fluid. So those are going to be our 3 steps of respiration, essentially how we get oxygen from the atmospheric air to our tissue cells, and also how we're going to get carbon dioxide from our tissues following metabolism and get that CO2 waste, get it out back into the atmospheric air when we breathe out. So first off, we have pulmonary ventilation. So in the pulmonary ventilation, air flows between the atmosphere and the alveoli of the lungs, and this is because alternating pressure differences created by contraction and relaxation of respiratory muscles is going to move air in and out. So we got our 2 steps of pulmonary ventilation being inhalation when we breathe air into our lungs, and then exhalation is breathing air out of our lungs. But again, this movement of air into and out of our lungs all has to do with alternating pressure differences. That's an important point there, and we'll talk about those pressure differences in the next couple slides. So we know that breathing in is called inhalation. We also call it inspiration. Now, just before inhalation, the air pressure inside the lungs is equal to the air pressure of the atmosphere. So this is right before inhalation. So air pressure inside the lungs. So atmospheric, I'll just put atmospheric P, which is atmospheric pressure. Now for air to flow into the lungs, the pressure inside the alveoli must become lower than the atmospheric pressure. Now in order to get that, this condition is achieved by increasing the size of the lungs. So to inhale, this is what we need. We need the pressure inside the lungs to be lower, less than the atmospheric pressure. Because if the lungs have a lower pressure than the atmospheric pressure outside of our bodies, that air is going to want to rush from areas of high pressure to areas of low pressure. Essentially, we can think of it the same way as a concentration gradient. Molecules are going to move from areas of high concentration or high pressure to areas of low concentration or low pressure. Now this brings us to Boyle's law. So Boyle's law talks about the relationship between volume and pressure. So the pressure of a gas in a closed container is inversely proportional to the volume of the container. So we'll get this right here. Volume of gas varies inversely with the pressure. So what this means is that if the size of a closed container is increased, the pressure of the gas inside the container is decreased. So if we have an increase in volume or an increase in size, we'll get a decrease in pressure. And the reverse is true as well. A decrease in volume of a container or a decrease in size is going to cause an increase in the pressure of that container or pressure inside that container. So if the size of the container is decreased, the pressure inside it increases. So we kind of get this inverse relationship between volume and pressure, and this is what Boyle's law is. Boyle's law just says that there's an inverse relationship between volume and pressure. And we can also see this with this demonstration picture on the right here. So here we have this volume. It says 1 liter here. So we have this piston that makes the volume or the size of this container about this large. When we have 1 liter volume, our pressure is going to be 1 atm, which is also known as 1 atmosphere. ATM is abbreviated for atmosphere. It's just a unit of pressure. So we can see in our pressure gauge here, we have it at 1. Okay, so this is a pretty good starting point. We have a liter of air in here, and the pressure is 1 atmosphere. And pressure is really just an indication of the force of the particles hitting the walls of the container. That's what pressure is. Pressure is essentially how many times these particles will hit the wall of the container. And so if the volume is very large, the particles won't hit the walls of the container that much. But when we make it smaller, the particles will hit the walls of the container more, which means the pressure is going to be more. So on the picture on the right, we decreased the volume. This piston came down. So now we're only this much volume, which is indicated here as ½ liter. So when we decrease the volume, these particles are squished in here now. They're going to be really vibrating quite vigorously, hitting the walls of the container a lot more. And our pressure doubles. And so here we have a higher pressure of 2. So if we have the volume, we double the pressure. We have that inverse relationship. So how does that kind of translate to our bodies? So if we want to get air into our lungs from the atmosphere, like I said, we have to decrease the pressure in our lungs so that the air is going to rush into our lungs from the exterior, because our lungs would have a lower pressure than the atmospheric air. And so in order to decrease the pressure in our lungs, or if we decrease lung pressure, how do we do this? So we decrease the lung pressure by increasing our lung volume. And we increase our lung volume a couple different ways. Our major way is we're going to drop the diaphragm, which increases the volume of our whole thoracic cavity, decreasing the pressure of our thoracic cavity, and therefore decreasing the pressure in our lungs. So on the left here, we have the muscles of inhalation. On the right, the muscles of exhalation. So kind of the first step in expanding the lungs to decrease that pressure involves the contraction of our main muscles of inhalation. So our diaphragm is going to be one of the main muscles of inhalation. And when we inhale, the diaphragm contracts and drops. And so we go from essentially a thoracic cavity of this size to when the diaphragm drops, our thoracic cavity can increase in size and thus decrease in pressure. When it decreases in pressure, we have air rushing in. Some other muscles that are involved in expanding the thoracic cavity are going to be external and intercostals. These are going to be the two main muscles, diaphragm with kind of some assistance with the external intercostals. These two muscles at the top, the sternocleidomastoid and the scalenes, help minimally, but our main muscle of inhalation is going to be the diaphragm with some assistance from the external intercostals. Okay? Now, when you forcefully inhale, you're going to use your external intercostals more. But during just quiet, regular inhalation, it's mostly diaphragmatic work. Now, on the other hand, when we exhale, we use a lot more muscles. So when we forcefully exhale, we're going to use our internal intercostals as well as our obliques, our external and internal obliques, and then our abdominis muscles, mostly the rectus abdominis and a little bit with the transverse abdominis. But you can see here when we exhale, there are a lot more muscles involved. Also, when we exhale, the diaphragm relaxes. We'll actually see the changes to the diaphragm a little bit more in the next slide. But it's mostly when we inhale that we're going to be contracting our diaphragm. So the most important muscle of inhalation is going to be the diaphragm. This is this dome-shaped skeletal muscle that forms the floor of the thoracic cavity. It's innervated by fibers of the phrenic nerves, which come from the spinal cord. And contraction of the diaphragm will cause it to flatten out, lowering the dome. So this increases the vertical diameter of the thoracic cavity. So when the diaphragm is relaxed, we kind of have this vertical diameter of the thoracic cavity. So this is relaxed diaphragm. And when the diaphragm is contracted, we're still going to start from the same part at the top, but contracted diaphragm flattens out quite a bit, and we get a much larger vertical diameter of the thorax. So that we're going to contract the diaphragm during inhalation, and the diaphragm will be relaxed during exhalation. So just a little bit more about the pressure differences during breathing. So during quiet inhalations, just kind of relaxed, calm inhalation, the pressure between the two pleural layers in the pleural cavity, which is called the intrapleural pressure, this is going to be always subatmospheric. So this is during quiet inhalation. Your intrapleural pressure is always going to be subatmospheric, which means that it will always be lower than the atmospheric pressure. But again, it's not by much. So here we have our atmospheric pressure, which is 760 millimeters of mercury, and our intrapleural pressure is 756 millimeters of mercury. But during those quiet inhalations, the intrapleural pressure is always going to be subatmospheric. Now, during more forced inhalation, the diaphragm contracts. You can see this here actually. Diaphragm contracts, and also the external intercostals contract. The chest cavity expands, and the alveolar pressure, the pressure inside the alveoli, is going to drop below atmospheric pressure. So here the alveolar pressure was right at atmospheric pressure, just during quiet breathing. But when we forcefully inhale, then our alveolar pressure is going to drop below atmospheric pressure. Now, airflow will flow into the lungs in response to that pressure gradient, and the lung volume expands. Now down here we have during exhalation, the diaphragm relaxes and the external intercostals relax. The chest and lungs will recoil, and the alveolar pressure increases above atmospheric pressure. So here we have a 762, and atmospheric pressure is still 760. See, it's not a huge change in pressure, but it's enough to force air out of the lungs. So the airflow will flow out of the lungs in response to this new pressure gradient. Not a huge pressure gradient, but like I said, it's enough to move air into and out of the lungs. So air pressure differences drive airflow during inhalation and exhalation. However, there are three other factors that affect the rate of airflow and kind of the ease of pulmonary ventilation. So these are surface tension. So this is surface tension of the alveolar fluid. The elastic recoil, and compliance. So elastic recoil, we can also call this airway resistance. So first off, with surface tension, we have a thin layer of alveolar fluid that coats the surface of the alveoli, the luminal surface, the interior surface of the alveoli, and it exerts a force known as surface tension. Now, this force must be overcome in order to expand the lungs each time. Airway resistance or elastic recoil. This is a factor due to the decrease in the size of the alveoli during expiration, which will increase resistance. Then lastly, compliance refers to how much effort is required to stretch the lungs and the chest wall. So these are just three other factors that affect pulmonary ventilation. The term for the normal pattern of quiet breathing is apnea. So this is just normal, quiet breathing. pattern of very shallow or chest breathing. This is known as costal breathing. And this consists of an upward and outward movement of the chest due to just contraction of the external intercostal muscles. Not much work from the diaphragm at all. So this is going to be just shallow chest breathing. And then a pattern of deep abdominal breathing is known as diaphragmatic breathing. Now this consists of the outward movement of the abdomen due to the contraction and descent of the diaphragm. So this is known as deep, also known as abdominal, or deep abdominal breathing is known as diaphragmatic breathing. Couple of other terms here, just to go over briefly. Apnea. This is temporarily stopping breathing, especially during sleep. So you may be familiar with sleep apnea. This is temporarily stopping breathing during sleeping. Dyspnea. This is difficult or labored breathing. And then tachypnea or tachypnea. This is abnormally rapid breathing. So some of the modified breathing movements that express emotion or in order to clear the airways are going to be listed here. So all of these movements are reflexes, like yawning or sneezing, but some of them can be initiated voluntarily as well, like crying or laughing. This can be initiated voluntarily. While at rest, a healthy average adult is going to average approximately 12 breaths per minute, with each inhalation and exhalation moving approximately 500 milliliters of air into and out of the lungs. Now we have this diagram here. This is called a spirogram. And this one in particular is going to work from right to left, with the start of the record on the right and the end on the left. When you do the respiration lab, though, your spirogram will actually work from the left to the right. So just to make note of that. So this spirogram shows someone taking in normal breathing for like one breath. See here, and then taking one large inhalation and one large exhalation, and then returning back just to normal breathing. Now the volume of one breath, just normal breathing, is going to be known as the tidal volume. So again, this is about 500 milliliters of just calm, normal breathing. Now, by taking a deep breath, you inhale a good deal more than 500 milliliters. So this additional inhaled air is known as your inspiratory reserve volume. So this is the amount of air that you can take in above your tidal volume. So kind of to the top of the graph that this person got for their forced inhalation. So essentially you breathe normally and then you take one huge breath and take in as much air as you possibly can into your lungs. And that top volume is going to be used to calculate your inspiratory reserve volume. So this inspiratory reserve volume is about 3,100 milliliters in an average adult male and about 1,900 milliliters in an average adult female. So again, this inspiratory reserve volume is the amount of air you can intake above your normal tidal volume. That inspiratory capacity, this is going to be the sum of your tidal volume and your inspiratory reserve volume. So kind of the total air that you can fit into your lungs is going to be that inspiratory capacity. When you fully breathe out and exhale as much as possible, get down to this volume right here. And so the amount of air that you can expel from your lungs beyond the tidal volume is your expiratory reserve volume. It's going to be approximately 1,200 milliliters in an average male and 700 in an average female. Now you're always going to have a little bit of air left in your lungs after you've completely expelled as much air as possible. You kind of don't want to have all the air leave your lungs. You still need a little bit of air to keep your lungs expanded just a little bit. And so there's always going to be this little bit of volume of air that cannot be forcibly expelled from the lungs, so that your alveoli remain open at all times. So that's where we get this residual volume down here. This is going to be the volume of air remaining in the lungs after maximum forceful expiration. So after you've expelled as much air as possible and you can't get any more air out of your lungs, there still technically is air in your lungs in order to keep the alveoli open, but you can't forcibly expel it. So that's where we get our residual volume from. And that residual volume can be calculated by using age and height, I believe. So you'll do those calculations in the lab. Now our functional residual capacity, this is going to be the sum of the residual volume and the expiratory reserve volume. We also have a couple of other terms here. Vital capacity is going to be the volume of air that you can forcibly inhale minus the amount of air that you can forcibly exhale. So you're going to take this volume up here and subtract this volume to get that total vital capacity. And then our total lung capacity, we're going to take that vital capacity and add our residual volume, the amount of air that's left in our lungs after we forcefully exhale as much as possible. So again, you'll be calculating all of this in the lab when you do your spirometry lab. The exchange of oxygen and carbon dioxide between the alveolar air and pulmonary blood occurs by passive diffusion. And this is governed by the behavior of gases as described by two gas laws. These are Dalton's law and Henry's law. So just very briefly going over these two laws that govern gases or the behavior of gases. So according to Dalton's law, each gas in a mixture exerts its own pressure as if no other gases were present. So the pressure of a specific gas in a mixture is called its partial pressure. And this partial pressure determines the movement of oxygen and carbon dioxide between the atmosphere and the lungs, also between the lungs and the blood, and also between the blood and the body cells. So this just means that even gases will move down their concentration gradient, but we're talking about a pressure gradient instead. So they'll move to areas of, like oxygen will move from areas of high oxygen pressure to areas of low oxygen pressure. Henry's law, this states that the quantity of gas that will dissolve in a liquid is proportional to the partial pressure of the gas and its solubility. So in body fluid, the ability of gas to stay in a solution is greater when the partial pressure is higher and also when it has a high solubility in water. Now compared to oxygen, much more CO2 is dissolved in blood plasma, and this is because the solubility of CO2 is 25 times or 24 times greater than the solubility of oxygen. Okay, so now let's talk about external respiration. So in external respiration or pulmonary gas exchange, this is the diffusion of oxygen from air in the alveoli of the lungs to blood in the pulmonary capillaries, and also the movement or the diffusion of CO2 in the opposite direction. So oxygen will move from the air to the pulmonary capillaries, and CO2 will move from the pulmonary capillaries to the air in the alveoli. External respiration in the lungs converts deoxygenated blood coming from the right side of the heart into oxygenated blood that returns to the left side of the heart. Now that left ventricle pumps oxygenated blood into the aorta and through the systemic arteries to systemic capillaries. Now remember that the exchange of oxygen and carbon dioxide between the systemic capillaries and tissue cells is called internal respiration. As oxygen leaves the bloodstream and goes to areas where the oxygen partial pressure is lower, which is the tissues, the oxygenated blood is converted into deoxygenated blood. Now, unlike external respiration, which occurs only in the lungs, internal respiration occurs in tissues throughout the entire body. So here we have a figure showing external respiration in the lungs and internal respiration in all other body tissues. So here we can see at the external respiration on the top of this picture, oxygen is going to diffuse from the alveolar air, where its partial pressure is 105 millimeters of mercury, and it's going to diffuse into pulmonary capillaries, where the partial pressure of oxygen is only 40 millimeters of mercury as it comes from the heart. So much, much lower partial pressure of oxygen in the deoxygenated blood that's coming to the lungs from the heart than the partial pressure of oxygen in that air coming in from the atmosphere. So that oxygen is inhaled and then diffuses into capillaries, bringing the partial pressure of oxygen in the oxygenated blood to 100 millimeters of mercury. Now, when oxygen is diffusing from the alveolar air into deoxygenated blood, then CO2 is at the same time going to be diffusing in the opposite direction. So here we have our partial pressure of CO2 in the deoxygenated blood is 45 millimeters of mercury, and in the air is 40. So even though it's a small difference, it's still going to be enough of a gradient to have CO2 diffused out and then exhaled. Now, in internal respiration, we're going to have the opposite happening. So here we have oxygenated blood that's coming back from the lungs. Our partial pressure of oxygen is 100 millimeters of mercury, and our partial pressure of carbon dioxide is 40 millimeters of mercury. Now, when it comes to the systemic capillaries and it gets to the systemic tissue cells, we can see that in the tissue, the partial pressure of oxygen is only 40 millimeters of mercury, so oxygen is going to very easily diffuse into the tissues. And then the partial pressure of carbon dioxide is 45, so 45 to 40 within the oxygenated blood, so it's enough that the carbon dioxide will diffuse out of the tissues into the capillaries. So again, just those differences in partial pressures of both oxygen and carbon dioxide will kind of determine the movement of these molecules. Now remember that oxygen does not easily dissolve in water, so only about 1.5% of inhaled oxygen is dissolved in blood plasma, which is mostly water. Most of the inhaled oxygen, the rest of it really, is going to be carried by hemoglobin in the red blood cells. Carbon dioxide, on the other hand, is mostly transported as bicarbonate ions. About 70% of carbon dioxide is transported as bicarbonate ions in the blood plasma, but it is sometimes also carried by hemoglobin in red blood cells when the heme group is not occupied by oxygen. Although the partial pressure of oxygen is the most important factor that determines the percent oxygen saturation of hemoglobin, there are several other factors that influence the tightness that oxygen will bind to hemoglobin, which is also known as the affinity of oxygen to hemoglobin. So here we have 5 factors that shift the oxygen hemoglobin curve. So an oxygen hemoglobin curve, which we will see in a second, kind of shows us the affinity that oxygen has for hemoglobin. So these are the partial pressure of carbon dioxide and the partial pressure of oxygen, the acidity or pH, temperature, BPG, which is another term for 2,3-bisphosphoglycerate, this BPG, this is going to be a substance that is found in red blood cells that helps to unload oxygen. As well as the type of hemoglobin. These are all going to affect how much affinity hemoglobin has for oxygen. or how tightly it binds. So this is an oxygen hemoglobin dissociation curve, and it shows the relationship between hemoglobin saturation on the y-axis here and the partial pressure of oxygen. Now this is at normal body temperature. So here, as the partial pressure of oxygen increases, so as we're moving this way down the x-axis, you can see the percent saturation of hemoglobin will also increase until it is saturated. So we get to this 100% saturation, where it kind of turns into a plateau. So this is at normal body temperature. So we can see down here when the partial pressure of oxygen is approximately 20 millimeters of mercury, this is going to be deoxygenated blood. So partial pressure of oxygen, if it's low, means that there's not very much oxygen. Then the saturation, or how many hemoglobin molecules are full of oxygen, is going to be quite low, around 35%. So we'll see this in contracting skeletal muscle, where oxygen is going to be released from the blood quite quickly to the skeletal muscle because the skeletal muscle is working and it needs a lot of oxygen to continue contracting. At a little bit higher partial pressure, about 40 millimeters of mercury, this would be in systemic veins, our saturation increases to about 75%. And then when we have this full 100 millimeters of mercury partial pressure of oxygen, this is going to be oxygenated blood, and this is in the systemic arteries. So this is just a general dissociation curve, oxygen hemoglobin dissociation curve, and we're going to see kind of how it changes as we look at those different factors that affect the affinity of hemoglobin for oxygen. So here we're going to look at some of those factors that influence the hemoglobin oxygen affinity. So on the left here, we're going to look at how pH affects the affinity of hemoglobin for oxygen. So our black line is a normal dissociation curve. Our blue line is a slightly higher blood pH, and our red line is a slightly lower blood pH, red being a little bit more acidic. So as you can see with the blue line with a slightly higher pH, or a little bit more basic blood, this causes the graph to shift to the left. When these graphs shift to the left, this is going to be hemoglobin having a higher affinity for oxygen. So we can see that it's a higher affinity because we can look at a single partial pressure. So we have 30 millimeters of mercury here. If we follow this line up to the blue line, at a higher blood pH, more basic, 30 millimeters of mercury is going to be about 65% saturated hemoglobin. Whereas the normal curve is going to be about right here, approximately 45-ish. So when we increase our blood pH at the same partial pressure of oxygen, hemoglobin is more highly saturated, which means that it has a higher affinity for oxygen with a slightly higher blood pH. When we look at more acidic blood or lower pH, this red line, this causes the graph to shift to the right. And this means that it will have a lower affinity for oxygen. That hemoglobin will have a lower affinity for oxygen. Because if we look at having 30 millimeters of mercury partial pressure of oxygen again, you can line this up and we're looking at about 35% saturation of hemoglobin. So at that same partial pressure, there's the same amount of oxygen in the blood. There's not as many hemoglobins that are bound to oxygen. So even if we just think of this, instead of partial pressure, we can just think of it, for example, as molecules of oxygen. There are 30 molecules of oxygen present in the blood. And of the 100 hemoglobin molecules that are also in the same area, when there's a low blood pH, only about 35 of those hemoglobin molecules are bound to the 30 possible molecules of oxygen. At normal blood pH, about 45% of the hemoglobin molecules are bound to oxygen, and we have a really high blood pH, or very basic, then 65% of the hemoglobin are bound to those same 30 molecules of oxygen. So in general, we can think of when this graph shifts to the left, the hemoglobin will have a higher affinity for oxygen. And when the graph shifts to the right, then hemoglobin has a lower affinity for oxygen. This graph on the right shows the effects of partial pressure of carbon dioxide on the affinity of hemoglobin for oxygen. Now with a low partial pressure of carbon dioxide in the blood, remember this is going to make the blood less acidic. Less acidic blood, also known as increasing the pH. The graph is going to shift to the left, meaning that with less partial pressure of carbon dioxide, the hemoglobin will have a higher affinity for oxygen. On the other hand, this red line, when we have a higher partial pressure of carbon dioxide in the blood, this is going to make the blood more acidic, or you can also think of it as a lower pH. This is going to cause our affinity graph to kind of shift to the right, making hemoglobin have a lower affinity for oxygen. So in this graph, we're looking at the effect of temperature on hemoglobin oxygen affinity. So again, the black line we see is normal blood temperature, about 37 degrees Celsius or 98.6 Fahrenheit. Now within limits, as the temperature increases, so does the amount of oxygen released from hemoglobin. So if we have increasing temperature, oxygen is released easier. Now as temperature decreases, the affinity for oxygen increases. So we can also think of affinity as how tightly or how strong hemoglobin is going to hold on to oxygen. So it's not going to want to release it. So here, as we see, as the temperature decreases, the graph shifts to the left, and so those red blood cells are going to hold on to oxygen more readily. As temperature increases, the affinity will decrease, and so the red blood cells release oxygen more readily. Now this makes sense when we think about how metabolically active cells require more oxygen. So like when muscles are working, they're going to produce a lot of heat and they require more oxygen. So as muscles are working, like you're doing a workout or running or something, the temperature of your muscles increases. And so we're going to kind of see you over here with the higher temperature. At least the temperature around muscles will be increased. And so as you breathe in oxygen, the red blood cells around the lungs will hold on to the oxygen pretty well because blood temperature around your lungs will be normal, closer to normal, but around your muscles, as that blood travels towards your hot muscles that have been working very hard, the temperature will increase. And so as the blood gets closer to the muscles, that increase in temperature will decrease the affinity hemoglobin has for oxygen. All that to say is that your red blood cells, or the hemoglobin within red blood cells, will hold on to oxygen until it gets to those highly metabolically active muscles where it's going to be hotter, and then it releases the oxygen really quickly. Fetal hemoglobin, which is often abbreviated HB-F, this also differs from adult hemoglobin. So adult hemoglobin, male or female, doesn't matter, but it's going to be abbreviated HbA. And fetal hemoglobin differs from adult hemoglobin in structure, but also, quite notably, in its affinity for oxygen. Fetal hemoglobin actually has a higher affinity for oxygen, and this is because it binds BPG less strongly. So it's going to bind BPG. Now recall that I briefly mentioned that BPG is a protein that, well, molecule, I guess, that helps to unload oxygen from hemoglobin. So it's going to help to unload that oxygen from hemoglobin. So if the fetal hemoglobin binds that BPG less strongly, then they have less BPG, and so they're going to hold on to oxygen stronger. They're going to have that higher affinity for oxygen. So when partial pressure is low, so maybe here at like 30% partial pressure here, fetal hemoglobin can actually carry up to 30% more oxygen than the maternal adult hemoglobin. Now this is particularly important because the oxygen saturation in the maternal blood and in the placenta is quite low. And the fetus might actually suffer hypoxia were it not for the greater affinity of fetal hemoglobin for oxygen. So it's really quite important that we do have this left shift of the graph for fetal hemoglobin. So here we have a summary of the chemical reactions that occur during gas exchange. Our top picture is going to be during external respiration, and our bottom picture is during internal respiration. So here, just to orient yourself, we have the pulmonary capillary wall. On the left is the alveoli, and on the right is the red blood cell inside the blood plasma. So we inhale oxygen. It's going to diffuse into the interstitial fluid and then cross into the blood plasma. And then it will diffuse into the red blood cell where it binds with hemoglobin. Now the binding of oxygen to hemoglobin will actually release hydrogen ions. So if we left these hydrogen ions unchecked, it would actually make the red blood cell and subsequently the blood plasma quite acidic. So we kind of need to do something about that. That's where this little molecule comes in. So HCO3, this is a bicarbonate ion. We know that the bicarbonate ions are buffer ions, and they again work as very important buffers in this reaction as well. So they will diffuse into the red blood cell, we can see here, and they will buffer those hydrogen ions that are released as a result of oxygen binding to hemoglobin. So when they bind to hydrogen, they become carbonic acid. So we know that H2CO3 is carbonic acid, and with the help of carbonic anhydrase, I can draw this right here with carbonic anhydrase, which is an enzyme, carbonic acid can very easily be converted into CO2 and water. So we can see this here, we have water and CO2. The CO2 diffuses out of the cell and then into the alveoli to be exhaled. CO2 will also come from the red blood cell itself, if it was carrying CO2 from the tissues as well. So we'll actually see where it gets CO2 from tissues a little bit later at the bottom picture here. But the hemoglobin can also just directly bind to carbon dioxide. And then when it comes here to the pulmonary capillary, it will also get rid of that carbon dioxide in the same way where the carbon dioxide just diffuses out into the alveoli. Now there's one other very important thing that happens here. So when this bicarbonate ion diffuses into the cell, it's bringing with it its negative charge into the cell from the plasma. Now because of this, we kind of have to have this equalizing factor. If we're bringing a negative charge in, we also need to send a negative charge out. We need to maintain electrical balance. So in order to maintain that electrical balance, there's going to be a chloride ion that's going to exit the red blood cell for every bicarbonate ion that enters. So you see this chloride ion leaves. This is called a reverse chloride shift. So again, this is just to maintain that electrical balance. Okay, now on the bottom figure, we have carbon dioxide diffusing out of tissue cells, diffusing into the interstitial fluid, crossing over that systemic capillary wall into the blood plasma. This is where a couple things happen. So CO2 can take this top route here, where it's going to bind to hemoglobin. Now this right here. This is a molecule known as carbaminohemoglobin. So when carbon dioxide comes in and binds to hemoglobin at the tissue level, this is how hemoglobin is transported in the red blood cell. Some of the carbon dioxide will come down here, and it will combine with water to produce carbonic acid and then dissociate into bicarbonate ions and hydrogen ions. So this is the same reaction we saw at the above picture, but just reversed. So in these cases, hemoglobin is already bound to oxygen. Hemoglobin brought this oxygen from the lungs, and when those hydrogen ions are released, hemoglobin will act as a buffer. So here, this is where hemoglobin acts as a buffer. And in this case, the hemoglobin will release its oxygen it's holding on to, and it will bind to the hydrogen ion. The oxygen releases, and because the partial pressure of oxygen in the red blood cell and in the blood plasma is much higher than in the tissue cell, the oxygen will very quickly and easily diffuse into the tissue cell. And then this bicarbonate ion right here is going to diffuse out into the blood plasma. Now again, to maintain electrical balance, we're going to also have this chloride ion move into the cell. Because we have this negatively charged bicarbonate ion leaving the cell, we're going to counteract that by having a negatively charged chloride ion go into the cell. This is known as a chloride shift. The size of the thorax is altered by the action of the breathing muscles, which contract as a result of nerve impulses transmitted from centers in the brain, and these muscles relax in the absence of nerve impulses. These nerve impulses are sent from clusters of neurons located bilaterally in the brainstem. So what this means is that these clusters of neurons are located on both sides of the brainstem, the right and the left. Now, there's a widely dispersed group of neurons that are collectively called the respiratory center, and these can be divided into two principal areas based both on location and function. First, we have the medullary respiratory center, which is found in the medulla oblongata. And then is the pontine respiratory group, which is found in the pons. The medullary respiratory center, this is involved more with the rhythmic and quiet breathing. Whereas the pontine respiratory group, this is more the part that modifies the rhythmic breathing, such as when you have to modify your breathing in exercise or talking or sleeping. This is all completed by the pontine respiratory group. Activity of the respiratory center can be modified in response to inputs from other brain regions, even receptors in the peripheral nervous system, and other factors in order to maintain the homeostasis of breathing. Now, because the cerebral cortex has connections with the respiratory center, we can voluntarily alter our pattern of breathing. So these would be our cortical influences. Voluntary control is protective because it enables us to prevent water or irritating gases from entering our lungs. So we can hold our breath underwater, or we can hold our breath if there's bad toxic gas in the area. So we can voluntarily control our respiration. The ability to not breathe, however, this is limited by the buildup of carbon dioxide and hydrogen ions in the body. Now, certain chemical stimuli can also modulate how quickly and how deeply we breathe. So these would be our chemoreceptor influence. We have chemoreceptors in 2 locations of the respiratory system that monitor the levels of carbon dioxide and hydrogen ions, as well as monitoring oxygen levels, and they provide input to the respiratory center as well. So the chemical stimuli that modulate how quickly or how deeply we breathe are detected by chemoreceptors that are located either centrally, found in the central nervous system, usually they're in or near the medulla oblongata, or they could also be found peripherally. So in the peripheral nervous system, these are usually found in the aortic bodies or the carotid bodies. So these are clusters of chemoreceptors that are found very, very close to the baroreceptors. Now, normally the partial pressure of carbon dioxide in arterial blood is about 40 millimeters of mercury. Now, even if a slight increase in the partial pressure of carbon dioxide occurs, the central chemoreceptors are stimulated and respond really vigorously to the resulting increase in hydrogen ions. Now, this is a condition called hypercapnia. If arterial partial pressure of carbon dioxide is lower than 40 millimeters of mercury, it's called hypocapnia. Hypoxia, on the other hand, is an oxygen deficiency at the tissue level. Now, often this is going to be due to high altitude or even obstruction of the airways or fluid in the lungs. Pretty much this is any time or condition where there's an inability to bring oxygen into the lungs. So here we have a homeostasis diagram or a negative feedback diagram depicting the regulation of breathing in response to a change in arterial partial pressure of carbon dioxide. This could also be decreasing pH, making the blood more acidic, or also a decrease in the partial pressure of oxygen. So the receptors that detect this are going to be those central chemoreceptors in the medulla and also the peripheral chemoreceptors in the aortic and carotid bodies. They're going to all send impulses to the control center, which is the respiratory group in the medulla oblongata. They're going to take this information, integrate it, and then send output to the effectors, which are going to be the muscles of inhalation and exhalation, to hyperventilate. So essentially, the control center is going to tell the effectors to breathe more forcefully and more rapidly to get rid of this excess CO2. So this will happen if there's excess CO2. This will also happen if there is excess hydrogen ions in the blood, or if there's a low partial pressure of oxygen. All those conditions will result in the effectors being told to breathe more rapidly and more forcefully. Move more air into and out of the lungs. So once we're breathing a lot more, hyperventilating, we're going to have a decrease in arterial blood PCO2, an increase in the pH, and also an increase in the PO2. This will return us back to homeostasis. This table here shows the stimuli that affect breathing rate and depth. So things like pain, irritation of the airways, blood pressure, these can all affect breathing rate and depth. So not really something to be tested on, but something just for interest's sake here. Now, the respiratory and cardiovascular systems make adjustments in response to both the intensity and duration of exercise. Now, focusing on how exercise affects the respiratory system, recall that the heart pumps the same amount of blood to the lungs as to the rest of the body. So, as cardiac output increases due to exercise, the blood flow to the lungs, which is called pulmonary perfusion, is going to increase as well. Also, the oxygen diffusing capacity, which is the measure of the rate at which oxygen can diffuse from the alveolar air into the blood. This may increase threefold during maximal exercise because more pulmonary capillaries become maximally perfused. So again, this definition of the oxygen diffusing capacity, and this is going to be a measure of the rate at which oxygen can diffuse from the alveolar air into the blood. With advancing age, the airways and the tissues of the respiratory tract, including the alveoli, become less elastic and more rigid, and the chest wall becomes very rigid as well. So the result is really an overall decrease in lung capacity. Age-related changes in the structure and functions of the lung can also contribute to an older person's reduced ability to perform vigorous activities, such as running. Now, just like any body system, the respiratory system is going to contribute to the homeostasis of all others. So in the cardiovascular system, The respiratory pump will help venous blood to return to the heart. With the urinary system, the respiratory system helps to regulate pH, the pH of body fluids by regulating blood CO2. The respiratory system is also going to promote the flow of lymph, again, just with that respiratory pump. It affects both the cardiovascular system and the lymphatic system, just to name a few here. And that will be the end of chapter 22.